By now, you should have learned two types of biotechnology that we utilize to studying the DNA, which are DNA profiling, both involving PCR and gel electrophoresis. The number two is genetically modifying organisms, okay, or GMOs. Now, the third technology you need to know is cloning. So let's start by defining the word clone. So what does it mean when you say clone? Maybe you're familiar with the idea of clone is a copy, as an exact copy of an organism. So the subject specific terminology that you need to know, the definition is clones are groups of genetically identical organisms coming from a single parental cell. So it came from one cell and it's genetically identical to this parent cell. Okay, we normally call that asexual reproduction. When an organism reproduces without the use of gametes or sperm and eggs, and this type of reproduction, asexual reproduction, is very common in plants. Okay. Um, However, it can also happen in the animal kingdom. So, for example, the case of identical twins, clone, they are twins. When the sperm and the egg meet, so you have a zygote, and this zygote normally divides and becomes many cells. Now, for the case of twins, the division of the cell is not regulated, wherein it completely divides and separates into two cells. And each of this will keep dividing and growing into individual embryos, but they originally came from one cell. So, in a way, that is an example of cloning. However, it's not really exactly cloning because for twins, although they came from one cell, we have we have found out that twins who are genetically identical, they still have different like fingerprints, pattern of fingerprints. So we normally just call them monozygotic, not really clones. We just say monozygotic, meaning they came from one zygote. Okay. Now, other examples of cloning in the animal kingdom is budding. Okay, in your hydra. Miss, what is a hydra? Hydra is it's a freshwater organism. It belongs to the phylum Nidaria, together with the jellyfish, and these are freshwater organisms that are about 1 centimeter big. Okay, so in the water they're green. They're this small, and this hydra they reproduce by budding. So as you can see here in the picture, okay, what seems to happen is that this seems to grow and then it buds off. That's why budding. So this is genetically identical to this. So this continues on to be another hydra floating in the water. So this is also cloning. Another example of asexual reproduction in the animal kingdom is your aphids. So this is an aphid. They are like your plant insects. Sometimes if you see a leaf, there's a really tiny, transparent, light green looking insect. We call that an aphid. And what's unique about the aphids is their ovaries, the cells in the ovaries can undergo two types of division. So it can undergo mitosis or meiosis. So sometimes it undergoes meiosis to produce a haploid egg that needs to be fertilized by a male aphid. Sometimes the cells in the ovaries just divides by mitosis. So the egg itself doesn't need a sperm anymore because the egg is diploid. Okay, again, there are cells in the ovaries can either undergo mitosis or meiosis. If the cells in their ovaries undergo mitosis, so that egg that underwent mitosis doesn't need a sperm anymore. So it can grow on its own and then hatch into a new insect. Okay, so those are some examples of cloning in the animal kingdom. Now, cloning is very common in the plant kingdom. Okay, um, this is when your plants grow into another plant from either their stem, their roots, or their leaves. So two examples that you need to discuss are, for example, for garlic plants. So imagine this. For example, I have a garlic bulb. So this whole thing. Put it in soil. So as I put it in soil, it's going to start to produce leaves. By the way, this is also a specialized leaf. Okay, so this leaf will start producing more leaves, your vertical leaves, and then these leaves, if there's many of the leaves, these leaves will undergo photosynthesis. And as the leaves undergo photosynthesis, the product of photosynthesis is more food for the plant. So the product of photosynthesis will be able to produce more garlic bulbs. So the garlic bulb growing its leaves, the leaves providing energy to form more garlic bulbs. So this garlic bulb is similar to this garlic bulb. Okay. Another example of cloning for plants are your strawberry plants. I don't know if any of you have been to Baguio. They have a strawberry field. So strawberry plants, they don't grow tall. They grow very short. They're a very short plant. And then they grow laterally. So, well, they grow both vertically. Hold on. Yeah, vertically. And then some of their stems grow laterally. We call them runners. They have lateral stems. So this stem, so a plant grows, right? Some of the stem grow laterally. Okay, as it grows laterally, it will, it can, the leaf can go under the soil and form roots like little roots. Then eventually form and grow another plant. So this plant is from this leaf, which is from this stem, from this plant. So this plant right here is identical to the plant here. This plant right here, the cells of this plant are identical to the cells of these plants that came from this parent cell. Okay, so you have to know these two examples of natural methods of cloning for your plants. Um, I don't know if you're familiar with gardening. Sometimes what our parents would do is they will have plant stem cuttings, like a stem of a plant, and then they put that in the soil, and then that plant will start growing roots and develop more stems or plants, which are just clone of that stem cutting. Okay. All right. So let's talk about the biotechnology now. That's just a background about how cloning is common in plants, and we have some examples of cloning in the animal kingdom. Now, cloning in biotechnology. So, there are types of cloning. There's two types of cloning depending on the source of the cell. So, the first one is we have an embryo. Whoa, 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 whoa. When we when we get our starting cell from an embryo, and then we clone it, or we get our starting cell from any body cell, and then we clone it. So we clone the embryo or we clone a body cell. Let's start with cloning an embryo. Embryonic cloning, that's the word I was thinking about earlier. Okay, so cloning embryos. So let's just say once upon a time, this cow and this cow, they're like the best in my herd. So what I do is I get the egg cell of this and the sperm cell of this, and I grow it in the lab. So I fertilize it in the lab. In the first few weeks of fertilization, it grows an embryo. Okay, so this embryo can grow into one cow. But what I do is all of these embryo are similar to each other. They all have the same genes. So what I do is I fragment them. In other words, I separate them into individual embryos. And each of this embryo is pluripotent, meaning it can become many cell. So there. And all of these new sub-embryos that I made, I put it in different surrogates. Surrogates is it's just a term for an organism that carries an embryo that's not its own. So I put it in these surrogates. And all of these surrogates will produce identical organisms. Sorry, my story is a cow, and then this picture this is a sheep. Okay, but anyway, all of these organisms from the different surrogates will be identical because they all came from an identical plate of an embryo. They all started as one embryo. We just fragmented, we separated all these individual cells in the embryo, all of which can become another organism. Okay? What's the problem with embryonic cloning? The problem with embryonic cloning is that, for example, I like the characteristics of this cow, so I mated this with this cow that I also like. But when their sperm and egg unite, there's shuffling of genes. I really don't know exactly what the characteristic of this offspring will be. So what's the point of making many copies of it if I don't know, I can't predict the actual characteristic of this embryo. I just know the mom is this, the dad is this, but the combinations might be different. So I don't know the characteristic of this embryo. So what we do is the second type of cloning, which is called cloning adult animals using differentiated cells. So we're cloning using body cells. Oh, okay. What is this type of cloning? This was popularized by this guy, John Gurdon. Okay. So what he did was he removed the nucleus of a body cell of a tadpole, okay, body cell of a tadpole, and then he used another cell, another egg cell, removed the nucleus of this. So this egg cell now has an empty nucleus. And then he used electricity so that the nucleus of this body cell will go inside the this empty egg cell. Okay? So now this empty egg cell contains the DNA of that body cell. So your body cell nucleus, you put it in an egg cell. So this egg cell now doesn't need a sperm cell, it's already diploid, and it will start growing into an embryo. Now the organism from this will have the exact same genome as my source of body cell. Okay? So it's like you get your skin cell, and then you get an empty egg cell, and you put your genome into the empty egg cell. If that grows, that will become another Messinina because it came from my body cell. It's my genome. It's an exact clone of myself. Okay. So to be able to understand that, you need to discuss somatic cell nuclear transfer as been done with Dolly the sheep. So you may be familiar with Dolly the sheep. It's the first cloned animal. Okay, and this is the story in cloning Dolly the sheep. So you have you actually have three sheep to think about, or you is a sheep. EWE, that's you. All right. So this was the original sheep that they want to clone. So they took a body cell from this sheep. So specifically, they took cells from the udder. That's the mammary glands of this sheep. Okay, and then they took another egg cell from a different sheep. So this is a Scottish blackface ewe. This was a Finn Dorset ewe. Okay, so they got an egg cell from a Scottish blackface ewe, and then this egg cell, they removed the nucleus of this egg cell. So this egg cell has no nucleus. Imagine just the cell with no nucleus. And then they introduce electricity. So electricity, and the the cells from the udder went inside this empty egg. The cells from the udder went inside the empty egg. In other words, the nucleus of this empty egg is the nucleus of the body cell of the sheep. Okay? And then what they did was, so egg was electricity. Um, that new egg cell with the body cell inside. Okay? They put it in a surrogate sheep, so another sheep that carried the embryo. So when this gave birth, the offspring or the sheep was identical to this sheep, the source of the body cell. This one is just the source of the empty egg cell. This one is just the carrier of the embryo. But this Dolly the sheep is exactly the same as this. Okay? So we call this technology somatic cell nuclear transfer. Somatic means body cell. So somatic cell, you're transferring the nucleus of your somatic cell into an empty egg cell. Okay? And the offspring will be an exact identical copy of the source of your somatic cell. All right. so this is so that's all for the third type of biotechnology that you need to know, cloning. Thank you for listening.